Welcome to the Research Like a Pro question and answer series. Today, Diana will talk about how to use GEDMatch tools for DNA analysis. And although she recorded this back in April and there's been a site redesign at GEDMatch in August of 2026, the tools are still basically the same functions, and so the video is still helpful. So hopefully you'll get some ideas about how to use GEDmatch for the one-to-one tool, one-to-many tool, and the segment triangulation tool as you use tools from GEDmatch as you do your DNA analysis in your genetic genealogy projects.
All right, so let's talk about GEDmatch tools. In our Research Like a Pro lesson, when we talk about tools, we introduce a few things, but we don't have time to really dig in much to any specific tool because GEDmatch has so many tools. So I thought it would be fun just to try a little bit of a process here and show you how you could use some of the basic tools to make actual progress on your research. So the ones we're going to talk about are the one-to-many and one-to-autosomal, which are both free tools, and the segment triangulation, which is a tier 1 tool. So you can use this for $10 a month and do all your work. If you decided this is going to be part of your research plan, then you could just pay for a month or two and then cancel unless you're going to keep using it repeatedly. So in one to many, we are going to find matches, and it's to identify our strongest candidates. So we're going to look at an example in a minute, and this is a good place to just get started. And then we have the one-to-one autosomal where we can take one of these matches that we find here that we want to work with, and we can compare the two kits. Because in GEDmatch, you have kit numbers and you can work with those kit numbers as if you're looking at their results, just like in Ancestry, where we want to have people share their DNA with us so we can look at their matches just like, they were ours. That's what GEDmatch does for us. With the kit number, we get to see all of their matches, at least on GEDmatch. And so this is the way when we do this tool, we're going to see how we can evaluate the shared DNA strength because we want to make sure these are real matches. These are real true segments that we're sharing, not false positives. And so this is a step that will help us with that analysis. And then if we find a good strong segment, we could take that and we could do some triangulation where we get to compare three different kits or even more. And this could lead us to confirming a shared ancestor and would give us some good evidence. So let's go through this little scenario here and see what it actually looks like on the website. Now, I did see a message when logging into GEDmatch a few months ago that they're redoing the website. And so, if you're watching this video later, it may look much different if they've completely done a redo. But for now, this is what it looks like. So we're going to start with that one-to-many tool, which is the first thing that you can start with. And we're going to use one of my key test takers for my project. And I've anonymized all the names here. We're going to call him Thomas. And so I put in his kit number here. And you'll notice that the centimorgan size, I have changed. The default for the centimorgan size for people uh, matching Thomas was at 7. And we know 7 is a little bit small for segments. We want to have something, I like to go over 10. In this case, I decided I wanted to go over 12. And you can adjust this to have your segments be much larger. So if you're trying to find people with the largest segment size that you're matching on, for any specific case that you're working on, you can certainly go adjust this, which is really neat because you don't it's pretty simple to do. Just type in your number there and then see what happens. And you can go back and readjust, try it all sorts of other ways as well. And one thing I wanted to notice, if you want to find people who just are matching on the X chromosome, you can also do that. So you can just leave the default for everything else, which is what I did. This was one I really wanted to look at. And of course, you can just play with all these and see what they mean and work on those. There's a little tip section. You can watch a video, but this is basically what you need to know right here. Just adjust this to what you want it to be. So when I did this on Thomas, I found that Thomas had a lot of different matches. And I noticed one right away who was, I'm going to call him Richard. And I noticed that I had put in notes for him. I had also found this match over on Ancestry, so I knew who he was. And I knew that for Thomas, he and Richard were third cousins once removed, and they are on my Royston Dillard line. So I am working right now continually trying to find the father of Cynthia Dillard. She married Thomas Royston. And so I'm looking for people that could possibly have shared segments or be in clusters, and that eventually I could use that information to figure out her father. So you can see here that he's a male, and here's the total centimorgans shared. And there's a lot, 118 centimorgans, and the largest segment is 29 centimorgans. So this is a good match. This would be a good one to look at to see what's going on here. There's a lot of DNA shared. There should be some possibilities for maybe some triangulation there eventually. So what do we do next? Well, I've got this match and I've got his kit number. So now I can switch over to the one-to-one autosomal. So now I'm going to just compare those two. So I could have picked anybody here to compare with Thomas, but because I actually knew who Richard was, I chose him. And so we put those two kit numbers into the tool, and you can just keep the default settings. You're going to see now a chromosome browser. So you have a default of showing you the graphic and the positions. So the positions would be just like a table. If you only want to see positions, you would just get the table of all the start and stop points, how much DNA is shared and all of that. And why would you want to just do that? Well, maybe you want to simply put all of that into your chromosome map on DNA Painter, and you just need the data. You don't want to have the graphics as well. So you could have that just show you the actual data, or maybe you just want to see the picture, see the graphics only, or maybe you want to have it combined. So you can also select display only chromosomes with matched segments. So I decided to try that out because there are a lot of chromosomes and I don't need to see all the ones that don't have any matching. And I decided to stick with the both graphics and positions. Now, there are some other things here that do we need to worry about this? Well, we always want to understand what we're looking at. And so we're going to see that there are different colors. When we get to the chromosome map, there's some colors. And so if we have got base pairs with a full match, this would mean if you're comparing siblings and they are matching on both the maternal and the paternal chromosome, then you're going to get a green. Most of our matches will just be half matches. They're just sharing with us on one side of the family. However, if you do have something, an unknown match and they show up with a lot of green, that's going to show you something interesting that you'll need to figure out how they're connected, on both maternal and paternal sides. And so we're going to usually see the yellow. Now, we also want to pay attention to this blue. And so we want to have darker blue or at least in the middle. And this just means how dense are the SNPs here. So we know that when the testing companies test someone's genome, they're not testing the full genome. There are some that are starting to do that now, but the tests that we're working with generally are just testing a certain amount of SNPs on each chromosome. And so it's just good. We're going to want to see, make sure there's a decent amount of SNPs that have been tested. So that helps us again to see if it's a valid segment. So we're just looking for that. We're looking for what's going on with the colors. And those are some things to keep in mind as we see our picture. So here we have the data and then we have the picture. We have the chromosome here. And so most of it, there's no match, all of this black part. But here is a match. It's yellow, so it means it's just matching on one side of the family. And we can see that it's got, a decent amount of purple there. So it must be a relatively dense SNP, or at least dense enough that it can provide, good data here on matching. Now, I like this number, 29.5 centimorgans. That's a good size segment. This is just a portion. I believe there were 7 different chromosomes that um, the two matched on. And so we're just going to look at this one right here. It's a good size segment. There's a good possibility that maybe I could find a triangulated segment because this is pretty large. So now we hop over to another tool, and this is the one that you do have to pay for, segment triangulation. And it is, like I said, $10 a month, or you can pay yearly if you want. And on this tool, you get to select which chromosome you want to look at. So here you can see this is chromosome 11. I want to look for some possible triangulated segments right here on, possibly one triangulating with this. And so I have again kept it at that 12 centimorgan. Let's just get rid of some of those really small ones. And when I do that tool, then I find that I've got a whole bunch of different segments here. Whoops. I have a whole bunch of different segments here. And here is one between Richard and Robert. And it looks like it's a triangulated segment. So what's my next step? Well, that would be to find the most recent common ancestor of those two. Now, I can also paint that segment data on a chromosome map on DNA Painter. So I have quite a few different segments here that are painted, but this isn't my match. This isn't my triangulated segment. This is the triangulated segment for Thomas. And so I actually have a chromosome map for Thomas, all of his own. And so what I would do, I would take that data and I could paint that onto his map. So we always want to make sure our chromosome map is for one person, for the key test taker, and see who all shares. But I can also look at that segment data and see if it overlaps with me and on that chromosome 11 and how close it is to anything that, any hypotheses I have. So GEDMatch, what does it do for you? It just gives you more information. And this is why we often want to have our testers or our DNA matches upload there because then we can use all of these different tools. Some of them, well, probably all of them, are not super easy to understand how to use. So my biggest takeaway is to play with them a little bit. Take your objective. What is your focus? What do you want to learn about? Take your own results or one that you have access to, a key test taker like I did with Thomas, and then use their kits to find relevant matches. Start exploring, start seeing if you can learn to use these tools. Because as you go further back on your tree and you get more and more difficult with trying to tease apart the DNA, and you have all these different ancestors that it could be with, could be descending from, you may have to go to segment data. So, learning how to analyze segments and how to work with these tools can be a really good thing to know how to do. And, we just all learn by experience and practicing. So, go try it out and see what you can figure out and see if it could help you. And the good thing is that once you do start to learn a little bit about it, you'll know how to put it into a research plan. If you don't understand a tool or have any ideas how it could help you, then you probably would never include it in a plan. So, as part of the Research Like a Pro process, we like to plan our research with our documents as well as our DNA. And that is very helpful for us because we got to know a little bit of something before we can put it in a plan. All right. So, a question on this. How much do I use or Nicole use GEDmatch for our work? We generally use just the clustering tools. We're usually using pedigree triangulation and looking at trees, trying to find the common ancestor of shared matches and that type of thing. That's what we do first. And if you remember we have a bell curve that shows what do you do first? Well, you do all the parts first that are pedigree triangulation and genetic networks. And then after that, you work with segments. But eventually you will come to a point where you may need to start working with segments. And so it's just something that we want to be aware of and see what we can do to learn more about it. I actually and thinking I probably will have to get to segments on my Cynthia Dillard case just because, which is why I'm starting to explore it and learn more about it. So there you go.
I will say too that I don't use it very much. And I did use it more when I was getting ready to publish um, one of my DNA case studies it was actually as a work sample in Research Like a Pro with DNA, the book. And so I wanted to get as many kits over to GEDMatch as possible because it's just a great place to put test takers kits so that other people can see their results if they want to verify the work that you've done. So when you're publishing, I think it's another time to consider using it. And of course, it's great to start with putting your test takers there because then you can get more evidence for triangulated segments and that kind of thing if that's going to be possible for your project. So I
Well,
like to consider it as an
uh,
option for if you're planning to publish something to get started on it from the beginning.
right. And another good thing to do with it is you may not have your test taker, let's say, go back to my Thomas. He's not on 23andMe. I'm on 23andMe, but he's not. So what if someone had uploaded their 23andMe data to GEDmatch? If they had and they're a match, I would be able to see that match, whereas I wouldn't ordinarily because he's not on 23andMe. So not all of our test takers are going to be on every single different testing company. And so if we put them on GEDmatch, then hopefully we'll pick up a few more matches that maybe we wouldn't. It's just another place to work. So,
Yeah, and especially for ancestry test takers, you can look at your list of matches and see which ones are from ancestry, and then you can get their
mhm,
segment data. You know, if
yeah,
you've especially if you've already figured out who they are. So if you're doing a chromosome map on DNA Painter, that's like a really good time to like use GEDmatch. So sometimes I'll go there for that purpose.
agreed. I probably use GEDmatch the most for my DNA painter chromosome map to get the ancestry data out of it. And that's been great because I know a lot more of my matches on Ancestry than I do in other places. And so being able to go to GEDmatch and get the chromosome data put into my chromosome map is how I've actually filled out so much of it. You know, a lot of this data that I showed here is from GEDmatch. Most of these people were on GEDmatch. I had asked them to be on GEDmatch or I had figured out who they were. And so that really helped.